I thank Roland and Becca for inviting me and Barry for organizing this conference to share some recent results, actually. And it's basically a long-time research which we did in several EU projects, and it could be summarized under the term analyzing and interpreting toxicogenomics data at the network level. Just found the right button. Yeah. So, first of all, I briefly introduce omics characterization of cellular systems. What do we mean with that? Much has been said already by the two speakers before, so I can be short there. The term pathway has been mentioned a couple of times before, so I want to introduce you to a tool that we developed in my group. So we are a bioinformatics group at the Max Planck Institute, working in genome analysis mainly. So it's a ConsensusPathDB, it's a pathway network resource. I will briefly show what you can do with that. Then I go into a use case which we did in the field of carcinogenicity testing in vitro assays, and point you to another resource which is the ToxDB, where we actually put forward the approach into a resource. And last but not least, I will try to show you the result of a pre-validation study, so which is the step after you establish a reasonable assay, and I want to show what we experienced there. So coming to the first point, Tanya mentioned that already, toxicity testing. I don't repeat, you all are aware of the importance of toxicity testing. There is a public need in testing all the chemicals and drugs before they get in contact with humans. And I just want to mention two things. So first one is that it has been shown already that toxicity pathways are quite different in rodents and humans. So that means we would need more human assays. And of course, we do not want to do that in vivo. We also want to spare animals being tested. So the goal is here for looking for human in vitro assays, actually. And this is what the topic is about. Then AOP is also mentioned before, and what particular points could add bioinformatics to these AOPs. AOPs start with a molecular initiating event, which is then associated with cellular processes, which can be measured, of course, and can be detected. So omics data, and Pekka gave you an introduction. Mainly these are microarray data, still in toxicology. So omics data could basically deliver you these key entities if you can associate them to pathways and phenotypic data. That means a whole lot of bioinformatics deals basically with the analysis of omics data. And the next step, we try to correlate this data and integrate these. And pathways, again, could be concepts where different omics levels could be nicely integrated. We try to identify networks, network modules, or individual pathways that are predictive for these specific hallmarks of toxicity that we look at. And of course, then we need to validate these networks in functional assays and integrate also chemical and physiological data. Why do we focus, or I said that pathways are natural data integrators, if you like, from one omics platform to the others. And this is a kind of motivating examples. Why do we deal with pathways? What you usually observe if you work with proteome and with transcriptome and then correlate those genes or proteins which are dysregulated in transcriptome and in proteome, you experience a very limited overlap. And this is an example from our research. So here we challenged a microarray with anti-cancer drug, in that case EGF receptor inhibitors, like cetuximab and erlotinib. So they're both doing essentially the same thing. They inhibit EGFR. Okay, so you could argue that these drugs should be quite similar. You know? And if you look at the transcriptome, how many differentially expressed genes do you have with cetuximab and how many do you have with erlotinib? Then you experience only a 37% overlap between these two drugs. So although they phenotypically do the same thing, it means inhibiting the EGF receptor, the transcriptome response is quite different. Okay? If you do it on the proteome level, here of course you have less proteins identified, so you have less numbers. This is a bit better, but still it's only 45% on the proteome level. If you look on pathways, actually, and you summarize the genes in pathways and see what pathways are dysregulated by the two drugs, you have an 80% agreement. That means the pathway is a concept which ensures a better comparability across different drugs, actually, which should do the same thing, and probably are thus closer. Such kind of analysis is closer to the phenotype, actually. So coming to a pathway, so once admitted that pathways are nice concepts, I want to introduce one of our resources, the ConsensusPathDB, which has basically a lot of pathways integrated. And this is a protocol actually from September this year, where we summarize all say the different analyses that can be done with the ConsensusPathDB. The main point is that we integrate 32 different public repositories, which is a main, which is really a large kind of resource for pathways. So you heard about CAG and Reactome in the talks before. We have those in there and many more. Biological pathways. This is a paper, a quite old paper, 10 years old, from Gary Vader's group. They built this resource called PathGuide, and at that stage already they found that there are 547 different databases in the web which agglomerate pathway concepts. So you can imagine there is a lot of information out there. So this points to the diversity of pathway annotation. We're not talking about anything like an ontology yet for pathways. Yeah? I wouldn't even touch that because it's so complicated. But for the ConsensusPathDB, we actually integrated 32 of the most common, if you like, pathways and interaction networks, and these basically cover different levels of molecular interactions, like protein-protein interactions, drug-protein interactions, gene regulations, and also pathway concepts, which means gene sets basically pointing to a phenotype, to a molecular phenotype. And why is that integration necessary? Suppose you have a pathway called apoptosis in 3 of the major databases, like Wiki Pathways, Reactome, and CAG. You could argue, well, I don't need 3 of them, I just take one of them. It's called apoptosis, it's the same phenotype. However, if you look for the molecules that are actually annotated with these pathways, they are largely different. So 70 of all the molecules annotated with pathways with apoptosis in these 3 pathway databases are actually unique for a specific pathway database, 70%. Yeah? That means working with gene sets and overrepresentation analysis with a specific list of genes that you find in your transcriptome analysis. You could, with one analysis you find a pathway significant, and with the other analysis you do not find the pathway significant. Yeah? That's what makes integration of pathway concepts really, really necessary. So the web server, what you can do with it, you can do this kind of overrepresentation analysis that you saw before. So where you actually ask, given a certain list of differentially expressed genes or proteins, you ask the database, give me all pathways that are enriched by this. You could also ask this by uploading own expression data and then do a kind of similar thing like gene set enrichment analysis, which is a more quantitative readout. And of course, you can look for single proteins and then, for example, generate a network environment which covers these different heterogeneous interactions. Now, I want to show you a certain approach that we followed, where we think this is superior to single gene analysis or single protein analysis, and this is quantifying pathways by omics data analysis. So how do we do that? So now we work only on this part of the ConsensusPathDB, that is the 4,000 pathways, the concepts that are integrated there. And this is the only slide with the formula. It's basically, an approach which has some side steps and so on, but essentially it's this. So what you have usually with your experiments is a gene over experiment matrix. You have a microarray and you have a quantitative readout for genes on your microarray for each particular compound that you're looking at. The database gives you a pathway over gene matrix, which is essentially a 0 or 1, saying this gene is annotated with that pathway or not. Now you simply make a matrix multiplication mathematically, and you have a pathway over experiment matrix, which means we can quantify actually the pathway given a certain experiment. And this could be transcriptomics, this could be proteomics, anything which is quantitative. Okay? Having that means we can, for each experiment, and here is an example of 15 compounds, 15 chemicals. Red are the genotoxic chemicals, blue are non-genotoxic chemicals, and green are non-carcinogens. What you have is actually, you have a different readout or a different quantification for each chemical on the pathway level. That means this chemical activates or changes a lot of pathways compared to this, for example. Another very interesting thing is, we heard before that dose is essential. So this pathway approach is actually sensitive of the dose. Yeah? So here you have pathways scored by this approach with gene expression data. And you have here a lower dose and here a higher dose. And what you see is a kind of shift. So this pathway scoring method actually reflects the individual dose, which is a kind of good thing because this is what we want to know. Is this pathway, if I give a certain higher dose to that particular pathway, then the alteration should be larger than with a lower dose. Okay? And we want to take now this approach, so this pathway scoring approach, and test it for hazard prediction of carcinogenicity. This was a paper we did within the ConsensusGENOMICS consortium in 2011, and we did that in human embryonic stem cells. And I would like to touch over this kind of use case with the next couple of slides. So what we are doing there, actually not we, but a company in Sweden. Now it's called Selectis. At that time, before, it was called Salatis. They developed a stem cell system where they basically differentiated human embryonic stem cells into an assay which is close to hepatic cells. Yeah? And they show that, for example, with several hepatic markers. Then, at several days of the protocol, they took RNA, and this RNA was challenged with 15 compounds of the 3 compound classes that we look at: genotoxic, 5 genotoxic carcinogens, 5 non-genotoxic carcinogens, and 5 non-carcinogens. And then they make an array, so microarrays out of it, and compared it against time-range controls. Okay, and our task was basically to build a kind of classifier which can distinguish the 3 classes. So we started with a naive approach, working on single genes. Okay, what you would probably do is in ANOVA, you have 3 classes, and you identify those genes as significant that came out with ANOVA. Okay? So if you do so, you end up with 592 genes. Okay, you have a lot of genes associated with metabolism, that's good. You even have genes that are associated with transcriptional regulation, that's even better, and you have tumor suppressors in there, changing and oncogenes, very nice. However, if you look at the discrimination potential of these 592 genes, what you observe is that you can basically very well separate the genotoxic group from the other ones. However, there is a poor discrimination between the non-genotoxic and the non-carcinogens. And that is basically the hard challenge that was not very sufficient. So we looked a bit deeper into the analysis. What we see is that basically we find 5 clusters, and what we saw is that the genotoxic response in these clusters basically dominates everything. Yeah? So the non-genotoxic specific genes didn't even have the chance to end up in that list of significant genes because the genotoxic were simply too dominant. Yeah? Remember what an ANOVA does, you have 3 classes, you look for the variance, and if the variance in one class is very, very high, usually this dominates everything. Okay, so gene approach for this. So what we then did, we used the pathway approach, the pathway scoring, which I described before. And what we saw there, we have pathways, like fast signaling, associated with apoptosis, for example, which is very dominant in the genotoxic. However, what we saw for the first time, which we didn't see in a single gene approach, we also have for the other groups, like in this case PPR signaling, pathways which are very, very specific for that particular class. And if we compare these patterns or this discrimination that we gained with the 592 genes, we did the same ANOVA approach with the pathway scores, and what we get is basically a nearly perfect separation with only something like 37 pathways. This demonstrates, in a way, that going from single genes to pathway concepts and then doing quantitative analysis with these pathway concepts is actually a more robust way and yields a better hazard classification for chemicals. We also observed that in other in vitro assays, like in this is a kidney system, which is a renal proximal tubular epithelial cell immortalized by the TERT protein. And also for the heparag, which is another liver assay. Yeah? So what we saw actually that in all cases, the analysis based on pathway concepts gave more clear results than on single genes. Okay, so having a large resource that gives us the pathways and having a numerical method to quantify actually these pathway responses from omics data, we have conceptualized this and built a resource which is called the ToxDB, and this is simply doing what we did before. So it was published last year within project diXA. It's again another new project. And what you get there is a combination of these pathway concepts with gene expression data mainly. And we use two big resources that probably all of you have worked on already. One is the TG Gates, and one is the drug metrics. They cover chemicals, drugs, and so on, tested on heart, liver, and other organs. And what we do there is basically screen all the microarray data, score the pathways, and then give the user a pathway view on the actually outcome of the exposure of the chemical or drug. And what you have is a compound view. So you have the chemical. You can select the experimental setup. For example, you choose drug metrics, heart, a certain dose and a certain time point, and then you basically get the most responding pathways out of this. So that's very simple, actually. And of course, you can always switch from the pathway to the gene view. So for example, clicking on one of these bars tells you these genes are in that pathway, and here is my fold change and my p-value, and what you see is, for example, that the pathway called striated muscle contraction is highly responding to doxorubicin, which makes some sense. And when you look in particular into the genes, then you see a lot of myosines being differentially expressed, and that's why it comes up. You always have the vice versa direction, so with pathways, suppose you are interested in a particular pathway because you have an assay that is questioning that particular pathway, like apoptosis, or in that case, mitochondrial gene expression. You could then again choose the study parameters, choose the study, the cell type, the dose, and the time point, and then you get all the experiments which generate a kind of high score for that particular pathway. So that is ToxDB. Because of time, I don't want to go further into that. Now, the last project I want to report on, and this probably comes closer to translational bioinformatics. Suppose you agree on a certain assay for hazard prediction or so. Then, of course, a big issue is the transformation of this assay. We call that an interlaboratory study. So that means the assay has to be shifted to other labs. It has to be functioning there. Data has to be generated, and so on and so on. And then the whole issue of interlab reproducibility comes up, and we were particularly interested in the bioinformatics part of that study. So what we designed was an interlaboratory pre-validation design. This was actually led by ECFAM. And what they did, they have blinded compounds. Nobody knows what compounds they got. We had two assays, one liver, one kidney, and they were grown in three different labs each, so 3 liver, 3 kidney. They generated the microarrays, the microarray data, and then we had 2 bioinformatic groups, so our group and Gene Data, which is a company which is dealing with toxicity a lot. And we had a lot of differences in our protocols. So we set up detailed protocols. So we had an SOP for bioinformatics, if you like, and they had an SOP for bioinformatics, and they were right. And the goal was first to assess interlaboratory reproducibility, and the second was to do classification of the compounds, because they were blinded. So we wanted to know whether it's genotox, non-genotox, or not. So, to make a long story very short, it was very striking that both analysis methods gave basically the same results. So, Gene Data did a kind of ranked approach, which give you a kind of information whether 2 labs generate similar outcomes, reproducible outcomes. And you see a case here where basically with the compound D, which we do not know, you see that the lead lab generated data which was found in the same way by the other two labs compared to random. That is a very significant result. And this is the result for kidney, where you can see that the lead lab with compound G has basically a very good correlation to this lab, but not to that lab. So you were able to identify an outlier lab, if you like. The same with our approach for the HePa-G. And we all found basically the same lab as an outlier lab for the kidney. But that means, for example, for these pre-validation setups, you need at least 3 labs. Yeah? We couldn't found that if we had only 2 labs. We wouldn't know what is the truth. So that was a story we learned. The other thing is based on reclassification of compounds, so gene data use. Basically, support vector machines with cross-validation. We used ANOVA, and again, we predicted the same thing. So we predicted the compound G for the kidney assay as genotox, E as non-genotox, A as non-carcinogen. And that was found in all 3 labs, even in the outlier lab. This is funny, because even if the data is not reproducible, it could lead to the same classification results. Yeah? This is something you should think about. But that's how it is. There are 2 different tasks. Yeah? That means you can classify the correct thing, but it doesn't mean anything. Yeah? But again, the two groups found the same results, and that was a very nice result of this particular pre-validation study. And this gives us actually some hope that bioinformatics generates stable outputs. Now, summary. So I showed you the ConsensusPathDB, which is a resource for pathway and molecular interactions. We developed a pathway scoring approach that used these concepts and measure omics readouts at the pathway level. I showed you with a use case on cellular hepatocytes in vitro assays that this can be actually used more efficiently for carcinogenicity, hazard prediction than a single gene approach. To conceptualize this, we have built a ToxDB, which holds these pathway concepts along with large omics data from published study. And I showed you a pre-validation study for in vitro assays, and that shows actually that bioinformatics approaches were fairly stable and gave similar predictions on interlab reproducibility, classification, and the identification of outlier experiments. I have something for outlines, so everything which I told you was based on microarray. However, this is fairly old technology. If you compare, for example, to the cancer field, people deal more with high-throughput sequencing. High-throughput sequencing is even going to, in cancer, to clinical practice by, for example, charting mutations of a certain patient and then predicting what drug should be. And mutations is, I think, not at all covered yet by toxicity, but it has a large effect. But this will probably come. We have published, together with colleagues in Maastricht, one of the first RNA sex studies actually for toxicology. And there, compared to microarrays, of course you have a gain in information, you have a knowledge on isoforms which you don't have with microarrays. You can look at structural variations and mutations, but however this all needs to be explored to a level that is as reproducible as microarray technology. And what we are also doing is, in a HeCaToS project, we are dealing now with epigenome data. So we nowadays you can basically sequence genome-wide methylation, which is an indicator for early changes. So transcriptome is more, say, second-level changes. And what we did, for example, we did a study on anthracyclines. So you have here doxorubicin, idarubicin compared to controls. And what you see is that the methylation level alone already gives you a nice prediction or nice separation of your experimental outcome. With that, I close. There were a lot of colleagues associated with all these studies, so I didn't make the exercise of writing all the names. Just I want to mention I thank the team of the carcinoGENOMICS, of the DIXA, and of the HeCaToS projects, which are all your projects, and I thank you for your attention.